Lipoproteins. This is Jack Brook from Columbia Gorge Community College. What I'd like to do in this session is talk about the four main lipid carriers that our body produces, and these are called the lipoproteins. So with our discussion of lipids, what you may and hopefully do remember is that lipids are not water soluble. They will float in water. With the bloodstream being mainly water, then we cannot just put lipids right directly into the bloodstream or else they would float. So in order to transport those lipids around the body, we produce lipoproteins that allow us to get the lipids from our digestive system to the body cells and to the liver and around your body. So looking at the basic structure of a lipoprotein, all lipoproteins have the same basic structure. They have a center core that is mainly triglycerides with some cholesterol esters, and I'll explain what a cholesterol ester is in a little bit. And then on the outside, the shell, kind of like a membrane surrounding the core, you have mainly phospholipid bilayer, or it's not a bilayer, but a phospholipid monolayer, similar to the plasma membrane. And then you have embedded cholesterol, and then you have identification markers that are called apolipoproteins. And for the different lipoproteins, you will have different identification markers so that the cells actually know what type of a lipoprotein there are. There are actually 5 lipoproteins, but there's only 4 that have any major importance in our bodies and in transporting lipids. And they are the chylomicrons, the VLDLs. IDLs are kind of minor. They're not considered one of the 4 major, LDLs and HDLs. And once again, the difference between these lipoproteins are going to be the amount of lipids they have in them and what type of lipids they have in them. So the basic composition is going to be the same, but the percentages of the components are going to be different. So this picture just shows you what a cholesterol ester is. A cholesterol ester is a cholesterol attached to a fatty acid. So we discussed fatty acids in our lipid section. Basically, when you have a cholesterol ester, that means that the cholesterol is inactive. It is not able to be used as to make hormones and to be, changed into something different. Cholesterol esters are for storage of cholesterols, and when they're being transported in a lipoprotein, most of them will be as a cholesterol ester. So if you can think of cholesterol esters as being the type of cholesterol that's inactive, and then when the fatty acid is removed and it's no longer a cholesterol ester, then it is considered to be free cholesterol, is considered to be able to be used to make hormones, to be used as a part of plasma membranes for new cells, and so it would be the active form of cholesterol. Now you'll notice that up at the top for the cholesterol with a Y, that's the way that it's officially spelled, but you'll notice in the picture it's cholesterol. So officially it's cholesterol ester, and then when it's free it's cholesterol. So we'll talk more about cholesterol esters as we go through. So I want to start with the biggest lipoprotein called the chylomicron. Chylomicrons are very high in triglycerides, and the reason is because you can see their origination. They come from the intestinal cells. So their job is to take the lipids that we eat. So mainly the lipids that are in food are triglycerides. They take those triglycerides, and package them as a chylomicron and transport those triglycerides from our intestines through the bloodstream to the liver. Chylomicrons are the biggest lipoproteins, 1,000 nanometers, and a nanometer is 1 billionth of a meter. And as you go through, what you'll see is that they are larger. It doesn't matter, that you know the actual number, but just know that chylomicrons are the largest lipoproteins. So they're high in triglycerides, they come from the intestines, and they transport lipids that we eat from food from the intestines on the way to the liver. And you'll notice that they do have two basic identification markers that identify them as a chylomicron. And the apolipoprotein is usually simplified just to be APO. So when you see APO, just think apolipoprotein. So the B48 and the E identification markers would be the two on a chylomicron. So here I just got a diagram that we're going to use in our discussion. And you'll notice that up here I have circled the chylomicron, and you can see here's the intestinal tract, here's dietary fat. So we're taking that dietary fat in the intestinal cell will actually form a chylomicron, and then it will be put actually first in the lymphatic system because it's too big to get into the through the walls of blood vessels. So it will actually go into the lymphatic system, and be transported until the lymphatic system actually dumps into the blood system later on. And so, but what you're also seeing, and I circled a chylomicron, then a chylomicron remnant. What you'll notice, number one, about the circle, you'll notice that this circle is bigger than this circle. And what they're trying to indicate there is that as the chylomicron goes through the blood system, this lipoprotein lipase, and it is an enzyme that one of its main function is to break down triglycerides that are in the chylomicron, and basically ultimately they be released. And as you notice, this arrow comes down and goes to a body cell. So you can see here it's taken up by body cells and made into triglycerides. So as the chylomicron is going through your lymphatic and blood system, if there are cells that need fatty acids, then the lipoprotein lipase, which is basically produced right there by in the cells around the blood system, will be released. They'll break down the triglycerides within the chylomicron, and then they will cause them to be released, and those fatty acids will be taken up by body cells, and then the cells can use them for what they want. So with that said, what that means is as the chylomicron goes through your bloodstream, it's going to lose triglycerides, and it's going to get smaller. So the percentage of triglycerides is going to be reduced based on need of your body cells. So when the chylomicron reaches the liver, it's actually going to be a remnant of its beginning self, and will be smaller, less triglycerides, and so forth. But it will enter into the cholesterol, into the liver, into the actual liver cells, and liver cells will actually take that up by phagocytosis, and we'll talk about that when we get into what we're going to call the receptor pathway in cardiovascular disease. We'll talk about that later. But in this session, I'm just introducing the type of lipoproteins. So that's the chylomicron. The next one is the very low density lipoproteins, or the VLDLs. They are also high in triglycerides, but you notice that the identification markers are changed. It's now has a ApoB100 and the E identification marker. Very low density lipoproteins, or VLDLs, are made by the liver. So you can see origination is in the liver. So the liver cells will actually package excess triglycerides, and then with the cholesterols and protein identification markers and stuff, The liver will package all of that into a VLDL, and then it will release that VLDL out into the bloodstream with the function of transporting those lipids. And so there basically would be excess lipids that the liver cannot handle. So the liver can only, it will take up the remnant chylomicron, basically break it down, release all its components, and then any excess triglycerides and then excess cholesterol that needed to make the VLDL, it will package those up, transport those lipids from the liver cells to the body cells to be taken up. So if we look at that, what I've circled down here is you can see the arrow coming from the liver. You make the VLDL goes into the bloodstream, but if you notice, we again have lipoprotein lipase that is going to break down the triglycerides, and the body cells are going to take them up. Now these body cells can be your normal body, like muscle cells or skin cells or whatever, but also excess fat would be taken up by adipose cells. So these cells could be your fat cells that are increasing. So the idea would be that the liver, everything that you eat basically goes to the liver. So all your carbohydrates, your proteins, and amino acids, those things, and your lipids are going to go to the liver. If you do not need those for body uses, those can be turned into triglycerides, into fat. And so you can get excess fat in the liver from eating too many carbohydrates, eating too many proteins, or eating too much fat. So all of those could eventually become triglycerides. Therefore, you would accumulate those triglycerides in the liver, and the liver would need to get rid of them. Well, the way the liver gets rid of them is to put them into a VLDL. And the VLDLs would then be transported to your body cells, and excess triglycerides then would be taken up by your fat cells, and then you would have bigger hips and a bigger stomach, basically, from eating too much food. All right. So then the job of the VLDL is to take the excess fat from the liver, transport it to your body cells. And then once you get rid of most of those triglycerides, what's going to happen is the VLDL is going to change its density and become an IDL. Okay. So an IDL is an intermediate density lipoprotein. All right, so what we're doing is the IDL is going to be a little bit heavier than the VLDL because the fat is going to make the VLDL a little less dense, and it's going to be lighter, if you will. But the composition of IDLs are high in cholesterol. They haven't lost their identification marker, so the identification marker is exactly the same as the VLDL, but they're smaller, and they are basically a remnant of the VLDL. So I need you to understand that the IDLs are not made by anything. The IDLs are a remnant of the VLDL. So the VLDL will lose its triglycerides, and because it loses and becomes more dense, then it will become an IDL. Now the function of the IDL doesn't stick around much because 2/3 of IDLs will be taken up by the liver, but that leaves 1/3 that can be changed into an LDL. So if we look at this picture, we can see here's the IDL down here, and then you'll notice one arrow goes up to the liver and one arrow goes to the LDL. And so 2/3 will be taken up by the liver, but 1/3 is going to become an LDL. All right, and there is an enzyme, and it doesn't matter what it is, but there is an enzyme that will become involved that is going to do that change of the IDL into the LDL. So basically, 2/3 will be taken up by the liver within about 6 hours, but there's going to be some leftover IDLs that will be changed by this enzyme into an LDL. Okay, so IDLs are kind of not considered one of the 4 biggies. As far as lipoproteins, kind of an intermediate type. So our next one would be the LDLs, and so these are going to be high in cholesterol. They, as you notice, they are going to lose one of the identification markers, the E identification marker. So this is going to be key, because now your cells will understand and be able to identify, yes, this is an LDL. It's not a VLDL, it's not an IDL, it's not a chylomicron, it's an LDL. Okay, and we will have these identification markers. They are going to match up with receptors embedded into plasma membranes. So it's high in cholesterol, and you can see it's smaller, 26 nanometers, but it's a converted by an IDL. So LDLs aren't really made by anything. I mean, LDLs aren't made by anything. Actually become, as it's changed by the enzyme from an IDL to an LDL. Okay, which kind of takes us back to the idea of, and we'll talk about it later, is how can you increase your LDLs? You can actually increase your LDLs by eating too much food. If you look at that relationship, what you would see is that if you ate a lot of food, and this is particularly fat, but anything, sugars and those kind of things, are going to go to the liver. If you have too much, the liver is going to turn them into fat. So the more fat that the liver needs to get rid of, the more VLDLs you're going to make. And the more VLDLs you make, the more IDLs you end up with. And the more IDLs you end up with, the more LDLs you potentially could have. So you can actually erase your LDL numbers, which we're going to see is the bad cholesterol, by eating too much food. Okay, I'm sorry. And then the LDLs, their job is to transport cholesterol to the cells. Okay, so LDLs are good. We need LDLs, but you may have heard of them as being bad cholesterol. Well, we'll talk about them in cardiovascular disease, and we'll show you that LDLs don't necessarily have to be bad. It's just when they're oxidized is when they're going to be bad. Okay, so as you see, the IDLs become LDLs, and then the LDL's job is to transport cholesterol to our body cells. So our bodies need cholesterol. If you've taken biology, you know that animal cells have cholesterol in the plasma membrane. Well, where do they get that cholesterol? They gets it from the LDLs. And then also what we'll see later is that if you have too many LDLs, that one of the jobs of the liver is to monitor homeostasis, if you will, of LDLs. And if the LDLs are too high, then what will happen is the liver will actually, through what we're going to call the receptor pathway, are going to take up LDLs by phagocytosis or endocytosis, and basically break them apart and reduce blood levels. So the main monitor of LDL numbers in your blood is the liver, and we'll talk a lot about that when we get into heart disease. But LDL's main job is to deliver cholesterol to the cell. So VLDL's main job is to deliver triglycerides. LDL's main job is to deliver cholesterol. So they're different, and we need both of them. The other one is the HDLs, and these, as we'll see, are considered the good cholesterol. And again, LDLs are bad cholesterol, because if you said to someone, a patient or something, well, you have bad lipoproteins, they wouldn't have a clue what you're talking about. or if you said, oh, you have high low density lipoproteins, they would have no idea. But everybody knows cholesterol, so they will just kind of simplify it and say LDLs are bad cholesterol and HDLs are good cholesterol. Not accurate, but it doesn't matter as long as you get the message across. HDLs are very high in protein. As you notice, they have one similar but another different identification marker, so that makes them enough different. They're very, very small, 12.5 nanometers. They made in two places. Your intestinal cells can make HDL and your liver can make HDL. And its main job is to transport cholesterol back for excretion, back to the liver, and to get rid of it from your body. And so it's going to mainly deal with LDLs. Okay, so we'll talk again about those when we get to heart disease, but I'm just introducing those today. So here's the HDLs right here, and you can see HDL transfers cholesterol for disposal. So they're considered the good guys, LDLs are considered the bad guys, and when we get into cardiovascular disease in the next session, I'll try to explain why. Okay. So just kind of an overall view of what we've talked about. You can see again the chylomicron is a huge son of a gun. And then we get the VLDLs, IDLs, LDLs, and HDLs. When you do get a blood test or a blood chemistry, sometimes they won't spell out VLDL, IDL. They'll just say what is the non-HDL cholesterol. Okay, so that's what it is. So the non-HDL would be these kind of together. And usually, you know, if you look at your numbers, if you've had a blood test recently, if you look at your numbers, a high VLDL number will usually translate into a high triglyceride number because they're associated. And then a high LDL number would associate with a high cholesterol number. Okay, and then a high HDL number would be hopefully, you know, have a relationship with a low cholesterol number, but it's not necessarily true. But we'll talk more about that as we get through here. I do want to show you that there are subclasses of lipoproteins, and we'll kind of refer to those as we talk, especially about the LDLs, because there are large LDLs, sometimes called large buoyant LDLs, and there are small LDLs. Small LDLs are considered the bad guys. They are considered the worst LDLs because they can become plaque in your arteries easier than the large LDLs from that. And then the HDLs, there are different amounts or different classes of HDLs, and probably I haven't read much about the differences between the classes, but what they're indicating here is that the size HDL2B is the one we want. Okay, so basically we will talk about these, but I just wanted you to know that they don't usually do routine tests on size of LDLs. The assumption is because of the way that we eat and our lack of activity and stuff like that, that they have to make that assumption that if you have a high LDL number that they're small. So I would guess in the next few years when they get a test that's economical, they may be doing tests to see how many small LDLs you have versus buoyant or large LDLs kind of thing. But they don't do it yet. You might be able to get it done if you requested it, but I don't know if they could. You may have to go to a bigger hospital or something that has the technology to do that. But anyway, lipoproteins do have subclasses, and some of the classes are increased risk for health more than other ones. And this last slide is just basically showing you the composition. As I mentioned at the beginning, all lipoproteins have the same composition, as you see up in the chylomicrons, as mostly triglycerides, but it still has phospholipids, cholesterol, and protein. And then the VLDLs, you can see it's smaller than the chylomicron, but it's still highest percentage is triglycerides. And then you can see the LDL highest percentage is cholesterol. And then HDL's highest percentage is protein. So both VLDLs and chylomicrons are very high in triglycerides, LDL's high in cholesterol, HDL's high in protein. But all of them have all four of those components. So those are all the basic components of a lipoprotein. So with this information, what we're going to do in our next session is talk about the relationship of lipoproteins to cardiovascular disease.